Hi, my name is Dr. Stefanie Chen from the Biotechnology program, and today we're going to go through an interim lab. As you can see, we're in the classroom today, so it's going to be a little bit different than your normal lab. The first thing you're going to want to do is find your lab station number. It'll be set up with everything that you and your lab partner need to complete this lab. The only PPE we'll be using today is gloves, so make sure you get a pair in your size. Your lab station should already be set up with your 3 plates from the transformation and ligation you did the week before, and your new culture tube. You also want to make sure that any personal beverages or food items are stored away either on the sides of the classroom or down by your feet, so they're not on the same surface as our bacteria. So the first thing you're going to want to do is count all of the colonies on your experimental plates from last week. So there are 3 main types of colony plates that you will encounter. So the first type of plate you'll encounter is a TNTC, or too numerous to count. This is when there's just way too many colonies to count, especially when they're touching each other, and it's just impossible to determine exactly how many there are. So this plate would be given a designation of TNTC. The second type of plate is when there are a lot of colonies that could probably be counted, but it would take you a while to count all of them. In that case, we do something called quadranting. You basically just draw a cross across the plate, and you only count 1/4 of the colonies. Then you multiply that by 4 to get an estimate of how many colonies are on the plate. The third type of plate is one where all the colonies can be fairly easily counted, and so you would just go ahead and count this plate. So one easy way to keep track of the colonies when you're counting would be to use your Sharpie and just dot each colony as you count so that you won't lose track. So the next step is going to be starting liquid cultures. And for this, you want to make sure that you only use your experimental plate. So pick out the one with your ligation and set aside the other 2 plates. So for this, you're going to use the toothpicks. You're going to take one out, being careful to be sterile, pick 1 colony, and then drop it in 1 tube of liquid culture. As you can see, there's 6 liquid culture tubes. So 5 are going to be your experimental colonies, and the 6th is going to be from a positive control plate provided by your instructor. Go ahead and get out a toothpick. And then you want to pick colonies that are fairly isolated and just streak across the top of the agar through the colony. And then take that and drop it right in the culture tube. So next we want to make sure we also inoculate a positive control. So for that, similarly, you're going to use your toothpick, pick a colony, and then drop that into one of your 6 culture tubes as well. So we only have culture tubes for 5 of our experimental colonies. Make sure you label all of your culture tubes with your station number and whether they're experimental or positive control. This will make sure that your cultures are returned to you properly during your next lab. So these liquid cultures will be used in your next lab session to purify out the plasmid and do a restriction mapping experiment to see how many contain the Campari insert that we want. Once you're done, all of your materials are going to need to go back on the cart that should be at the front of the classroom. So your 3 experimental plates from last week should be taped back together. After putting all of your materials away, you'll want to get out a bottle of ethanol and some paper towels and wipe down your desk area. So the last thing would be to remove your gloves and put them in the biohazard so we can begin class.